In the last lesson, we had talked about the biochemical characterization of the transforming principle, which was proposed by Griffith in his experiment with the mice and Streptococcus. And it was very well proven. You got an idea that it is DNA, but this scientific world and all the universities and all the scholars, they need a proof that cannot be questioned to a certain extent. So there was a need for a proof which made it quite certain that it was DNA only. Still, the protein standard somehow as a contender to the genetic material with DNA. So an experiment was conducted which proved that it is DNA only, and protein was thrown out of the race of this being a genetic material. And what was that experiment? That was done by Hershey and Chase, two scientists, in 1952. Both of them had done great deal of study in virology and bacteriology. So what they had to prove was that it was DNA and not genetic DNA and not proteins which were the genetic material. So as the two tough contenders, they chose that let there be whatever explanation comes, it comes from bacteria and virus. Now, why bacteria and virus? You know, virus contains only two things. One is the genetic material that could either be DNA or RNA. So it contains the nucleic acid. I am not going to write genetic material over here. Virus basically contains the nucleic acid and it contains protein coat. That is all. Okay. So they are going to choose bacteriophages. Now bacteriophages contain a DNA and protein coat. Right. Now they choose the bacteria E. coli for their experiment, and they choose a bacteriophage. Now you know the structure of bacteriophage, what it is like. It has a protein coat capsid like this, and it looks like a weird spider. To me it looks like this. This is the protein coat, and herein lies the DNA, right? This is a bacteriophage. Now you have to use this structure for understanding how out of these two, protein coat and the DNA, which one is carrying the information. Okay. So when the bacteriophage infects the bacteria, it sends its genetic material inside the bacteria. Bacteria considers that genetic material as its own and helps in replication of the virus instead. This is what is the work of bacteriophage. This is how it infects. Now what it does is, we know that it does is it sends the genetic material inside the bacterial cell, and this protein coat stays out. But it had to be proven at the time when this contest between DNA and protein was going on, which one is genetic material. These two people, they carried out an experiment where they used E. coli and bacteriophage. And what were the various steps which were involved in this experiment, that you have to keep in mind. And while we are doing those steps, you have to see how DNA stands out as the genetic material. And also I want you to pay attention to the minuscule details of this experiment which establishes that it is DNA only. Like the biochemical characterization was little bit vague in its explanation, this is going to be highly specific in explaining that it is DNA only. Now how? See, they are going to incubate, or they are going to grow the bacteriophages, which is the virus. Few of the bacteriophages would be grown on radioactive sulfur, and few of them will be grown on radioactive phosphorus. Now this was carried out. Why it was carried out? You know that DNA contains phosphorus. In the polynucleotide chain, it is the phosphate that comes and binds the nucleotides. So it contains phosphorus. Those bacteriophages which are grown on radioactive phosphorus will have their DNA getting radioactive. phosphorus was radioactive, DNA got radioactive. Now the sulfur, in case of sulfur, sulfur is present in proteins, but it is not found in DNA. So when the radioactive sulfur is taken into consideration, we have the proteins which become radioactive. Okay. So we have heavy atoms of both sulfur and phosphorus taken into account. The medium contains radioactive sulfur. Whichever viruses grow out of that medium, they will have radioactive proteins, and this will have radioactive DNA. Now, subsequently, we are going to conduct an experiment in 3 stages, where the first stage involves growing it on radioactive sulfur and phosphorus individually. The second stage is, first stage would be infection of bacteria, that is E. coli, with such 2 different types of viruses. Then they will be blended, and thirdly they would be centrifuged, right. Now we have to establish which one is the genetic material. We know that the information would be sent in and something would stay out. Now supposedly, if it is DNA which stays out, then we are going to see these 3 steps happening and how it happens. I hope you remember the name of these 2, the year when they worked on this. Let us take the consideration where we have radioactive sulfur. When there is radioactive sulfur, then we are going to have bacteriophages having radioactive protein coat, right. When there is the case of radioactive phosphorus, this would come here and this would come here, then you can when we have the case of radioactive phosphorus, then in that case protein coat stays normal while the DNA becomes radioactive. The next stage is blending these, and in blending we see that supposedly this is E. coli. This would come in contact with this particular bacteriophages. They will come in contact with E. coli. They would be blend, and they are going to send the genetic material inside. As a result of blending, this is the E. coli that we have. Okay. E. coli cell. Now what will happen when the infection takes place? They are going to send the genetic material inside. Now over here what happens in the next stage is that the DNA comes inside. It is not radioactive. This is E. coli's own DNA. Over here radioactive content gets inside the E. coli cell. That means when you find radioactivity inside the cell, that means the genetic material has gone inside. Now the next step would be centrifugation. Inside the test tube what will happen is that now the protein coat will get separated. Once it has been centrifuged, it will get separated and it will float as supernatant. In the case of protein, you are going to find radioactive supernatant while the heavier E. coli cells settle down. Okay. So the supernatant has radioactivity, that means it has not entered the cells. The infection has gone inside the cells as a result of blending, but what was radioactive stays above. That means the protein stays outside. What goes inside is the DNA. When you find radioactivity present in the precipitate E. coli cells, they have radioactivity, and from here it was deciphered that ultimately it is the genetic material which goes inside, and what it is? It is DNA. Alright. So this was an unequivocal proof that DNA is the genetic material. Now you can't question that it could be protein. Here it has been totally proven that it is not protein. It could be anything else, but at least it is not protein. Why it is not a protein? Because protein has stayed above. They made the protein radioactive. The radioactive protein could not get inside E. coli. Now because it is not getting inside the E. coli and infection has taken place, that means whatever has gone inside is certainly not protein. Then what has gone inside? It is DNA, where in the case of radioactive phosphorus, the radioactivity shows in the precipitate instead of supernatant. That proves that genetic material is nothing else but DNA, and it was proven by Hershey and Chase experiment.